Good afternoon, everyone. And today we have a great pleasure to have Professor Irina Gay Gaynanova from Department of Biostatistics at the University of Michigan. Before joining University of Michigan, Irina was faculty in the Department of Statistics at Texas A&M for 8 years. Irina's research interests include data integration, machine learning, and high-dimensional statistics, motivated by challenges arising in analysis of multi-omics data and data from wearable devices. Irina's research has been supported by the NSF, NIH, and recognized with a David Bayer Young Investigator Award from the Biometrics section of the ASA, NSF Career Award, IMS Delta, and Marvin Zella Emerging Women Leader in Data Science Award, and COPS Emerging Leader Award. Irina is also a dedicated educator. She deeply cares about the training of next generation and puts large emphasis on reproducible research practices and computational skills in her teaching. Her efforts in teaching have been recognized with Dr. Judith Edmiston Mentoring Award from the Texas A&M College of Science. So today, I think Irina is going to tell us something about distributional learning for wearable devices. Without further ado, let's welcome Irina.
Well, thank you so much for this very nice introduction. It's my pleasure to be here and to talk about my passion, which is continuous glucose monitoring data. And as Juhua said, thank you for a very nice invitation. And if you have any questions and you want to ask them in the middle, feel free to do so. I don't want you to get lost. And sort of kind of to start the stage, I work primarily with continuous glucose monitoring data. Those are the top two figures, and I'll tell you more about that and give you a scientific background. But in general, we have a lot of wearable devices these days. A lot of us are wearing Fitbit or Apple Watch, which allow us to track heart rate and physical activity data. And this technology sort of continued developing. We can track blood pressure. And because we can track a lot of the signals with high frequency over time, there is a lot of interest in using this data for inference, for classifying and finding phenotypes, and many other tasks. So I'm going to talk specifically about continuous glucose monitors today, but I think a lot of the work will generalize to actigraphy data or heart rate rated data as well. So to start talking about glucose monitors, I want to give you a little bit of context about diabetes, because this is where those monitors are primarily used. So when we eat food, it usually has carbohydrates in it. Some has more, some has less. And these carbohydrates, our body changes to glucose, which then we need to process for energy. And this processing happens in pancreas, which produces insulin, and insulin helps convert glucose into energy for our body. And in diabetes, this process gets disrupted. So in type 1 diabetes, your pancreas stops producing insulin altogether. So you have to take extra insulin to help process all this glucose. And in type 2, you have insulin resistance, which means even though you may still produce some insulin, you can't use it effectively for controlling your glucose. And so what happens is your glucose levels go too high. And it's a very prevalent public health burden because there is around 800 million worldwide with diabetes, type 1 and type 2. Type 2 is more prominent, and the prevalence just continues to rise. So why does it matter? And why this data is so complicated? Well, let's start with thinking, what does the normal blood glucose looks like? And that is, if you don't have diabetes, your glucose will be in the range of 70 to 120, roughly. These are absolute numbers. These are milligrams per deciliter. They mean the same thing across people. But your glucose is not constant. So as you eat food, your glucose spikes. It will spike depending on what you eat. If you have something very high carb, processed carb, like you got a pizza, you had a bigger spike. If you had like cheese, which has no carbs, you wouldn't have a spike. And so your body like recognizes the spike, produces the insulin, and your glucose goes down. So you have sort of this idealistic glucose profile over time where you have your food, you spike, you go down, you have your food, you spike, you go down, and so forth. It's very nonlinear trend. It's highly dependent on the environment, not just when you eat, but what you eat. And there's a lot of other factors that affect glucose. So your exercise, your stress, your sleep, if you get sick. And before the wearable technology has developed, sort of the standard way for us to assess what someone glucose is what's called this hemoglobin A1C or HbA1C. So if you go to the doctor, they will probably have it as one of your labs once a year. It's just one number that represents your average glucose levels in the preceding 2 to 3 months. And as a statistician, you can see that even if your one day looks like this, sort of summarizing that one day by an average is already sort of reducing a lot of information. And what you can also see is because the glucose is changing and reacts to so many things, this one marker, which is just one number representing an average, is probably not enough. So this is where continuous glucose monitors comes in. So they are very small devices. So this is a Libre monitor. This is a Dexcom monitor. They have a little tiny needle in them that goes in. You apply them like a stapler. So it's very easy to apply. And then they measure your glucose levels every 1 to 5 minutes. For Libre, the monitor usually lasts 14 days. For Dexcom, it's 10 days. So you wear one. When your 14 days go out, you trash it, you take a new one, staple it in, and it keeps measurements. And for people with diabetes, it's usually synced with an app on the phone, and that gives you real-time values. I usually work with data retrospectively, and so it collects it every 5 minutes typically, and then most recent CGM is Dexcom G7, and then there is Freestyle Libre Free. They are most popular. So to give you an idea what this data looks like, this is an example of the data from one subject who has type 2 diabetes. So here you have roughly 10 days. And it looks like line, but it's actually 5-minute frequency. And what the horizontal lines indicate is 70 milligrams per deciliter and 180 milligrams per deciliter. So for a person without diabetes, you would usually stay 70, 120, 70, 150. You can see that this person has higher peaks because the glucose is not as well controlled. And this horizontal lines here indicate 70 to 180, which is standard target thresholds for people with diabetes. And I will go back to those thresholds later on. So hopefully this gives you some idea. Very non-stationary, lots of peaks. We don't really know what happened here. We don't know exactly when they ate, what they ate, have they exercised. So this is the data that we're looking at. And again, just to summarize, the data can look very different from day to day and from subject to subject. So here I'm actually picked 2 subjects with type 1 diabetes, 1 day of measurements where I matched those 2 profiles by the mean values. So the mean value is exactly the same, but you can see clearly that the peaks are different, the range of numbers are different. And so mean is a very crude measure. So how can we summarize the data and how do we assess variability? And what I'm going to talk about today is specifically using distributional representation of this data, sort of looking at the distribution of all glucose values in this profile. And I will have 2 parts to my talk. First, I will discuss the project where we use this distributional representation to try to find what are the optimal thresholds, meaning what are the thresholds of glucose values that we should focus on and compare profiles in terms of. And then in the second part, I will talk how we can uncover the associations between the glucose measurements and other covariates of interest, such as medications, for example, or demographic characteristics using the distributional representations. So those are two separate projects, but the connecting part is CGM data and taking advantage of the whole distribution rather than just looking at the mean. So in search of CGM thresholds, and this is very recent work from our group, it's just been put on archive in January, and I want to acknowledge Jun Young, the lead on this work. He's an amazing postdoctoral researcher working with me, and also Neo Kok, who is a master's student at the University of Michigan and did a lot of work on data analysis here. So to kind of give you more specifics, when we have this high frequency wearable data, it's very common for clinicians and in general in analysis of this data to summarize it using fixed thresholds. For example, when we look at the actigraphy data, we often summarize it in terms of sedentary activity or light activity or moderate activity or vigorous activity. And the way it's done is you have certain thresholds, for example, for activity counts. You look at your actigraphy data over this time interval, and then you look what is the percentage of time that people are below a certain threshold, that is percentage of time they're sedentary. What is the percentage of time they're in this range, which is what is the percentage of time they're in light activity and so forth. And similarly for the CGM data, which is my primary motivation, there are clinical thresholds that are often used to summarize this data, in particular the 70 to 180 that I mentioned above. So looking at this profile again, so I have horizontal line for 70, I have horizontal line for 180. And the way this data often summarized and presented to the patients is we're looking what is the percentage of time that someone is below 70, between 70 and 180, and above 180. And then this percentage of time, also known as time in range, time between 70 and 180, is a metric that is often used for diabetes management, for treatment adjustments, for downstream tasks, such as comparing drug versus placebo in terms of glycemic control and so forth. So going a little bit more further into the thresholds, I know it's a lot of scientific context, but it's really motivating what we're going to talk about. There are these thresholds for CGM are consensus. In other words, there's an international consensus statements where everyone agreed these are the numbers at which we're going to threshold. And in range is 70 to 180, you presented in green. Hyper or high glucose is sometimes separated into level 1 and 2, where an additional threshold of 250 is introduced. Hyper or low glucose is also often separated into 1 and 2, where the additional 54 threshold is introduced. So you can either summarize this data using this 1, 2, 3, 4, 5 number summaries that sums to 100%, or you can combine the levels and then you have just 1, 2, 3 summaries that sum to 100%. And the typical treatment goal is to increase the time you spend in range where your glucose is not too high and not too low. Too high is bad because it's nerve damage long-term. Too low is bad because you can lose consciousness and it can even lead to death. So you kind of want to stand in this middle range. And then if you don't have diabetes and we're wondering where you are, there's a recent paper that find out that among people without diabetes, you are typically in range 94% of the time. All right. So this is now where we get a little bit more back into stats. How do we know that these numbers are optimal? What does it mean for these thresholds? And by these numbers, I mean 54, 70, 180 to 50 to be optimal. When these thresholds were designed, they were designed primarily for population of type 1 diabetes in mind. This is still the population that primarily uses these devices. As the devices get cheaper and more widespread, we're starting to use them for people with type 2 diabetes, with people who have diabetes during pregnancy, for pre-diabetes individuals, people who are interested in weight loss are sometimes interested in using CGM to see how their food affects their glucose and so forth. Should we use the same thresholds across different populations? What are the optimal thresholds? So our goal in this work was to define the threshold optimality as well as methodology for establishing data-driven thresholds for specific populations. And kind of go back to that this is not just an imaginary task. There is evidence in the literature, and here I'm looking within specifically glucose literature, but for the actigraphy, you can make the same argument, is that there is evidence that those thresholds should change. For example, in diabetes, rather than using 70 to 180, they use 70 to 140 during pregnancy, which is tighter. So the threshold is adjusted already here. Furthermore, there's the recent work by Kata that demonstrates that adjusting the thresholds could have different interpretations of treatment effect. In other words, the choice at which threshold you decide to summarize the data can affect your statistical power and downstream inference. And then you can use the full distributional representation, that is, rather than thresholding at 50, 70, 130, you look at the whole distribution of glucose values. And this is the motivation for this work. And then I'll show more how to use the full distribution in this paper that I'm going to discuss next. But there is still this part that when we do threshold-based summaries, they're simple. They're simple to explain to the patients. They're simple for clinicians to use. We have a wide range of standard statistical tools we can use with scalar summaries. So we're trying to see if we can basically still do the simple intuitive summaries, but do them better. And there is recent work by Banker et al. On data-driven thresholds with actigraphy data rather than CGM data, but it's focused on a supervised setting that is optimizing thresholds for specific outcomes. In this talk, I will focus on unsupervised threshold determination. So we do not have any outcome to consider. We're looking at the data itself, and we're trying to figure out what the optimal thresholds are. So this is how we're going to view this problem, and this is where more statistical part come in. So we have this high frequency signal from the wearable device data, and we use threshold-based summaries for that signal. In particular, within the CGM, we either use a 3-number summaries, high in range low, or 5-number summaries where we subdivide high and low into levels. These summaries always sum up to 100%, so you can think of it from the compositional data perspective, where low in range high is a 3-number summaries on a simplex, or if you use levels, then you have a 5-number summaries on a simplex. And the way we are going to think of the problem of threshold selection, that is selecting of the specific numbers at which we create this composition, is as the problem of histogram amalgamation, where amalgamation is combining. So to illustrate your point, when we look at the original distribution of CGM data, so CGM measures glucose as integers, so you have 70, 71, 73, and the device has a bounded measurement range because your glucose can only be bounded, and typical CGM measurement range is 40 to 400. Your glucose can actually go above 400, but most devices only measure within this range. So when you look at this whole profile, you could think of the empirical distribution where you know exactly how much you spend at 40, at 41, at 42, at 43. You can look at all of those individual numbers and look at this individual empirical distribution or histogram of the whole values that you observe. When we think of these threshold summaries, in particular, when you look at the consensus thresholds, which is 54, 70, 180 to 50, you can think of it as equivalently as summarizing this histogram by creating those cutoffs of thresholds that are shown here vertically. And instead of reporting individual percentages of time you spend at each integer, you report the percentage of time that you spend within this region. So by looking at those, you can think of it as you're combining the bins corresponding to the region that you have selected with those thresholds. Mathematically, if you think of a regional histogram, a regional empirical distribution as a compositional vector, where you look at all the possible values that your meter can take, 40, 41, up to 400, then by picking a threshold, you're basically combining the frequencies corresponding to the region as determined by the threshold. So you can think of your original empirical distribution as a very large simplex on 360, because that's how many unique values you can take. And then your consensus threshold gives you a reduced representation where you have only 5. And then for any given threshold, so if I were to pick a different threshold, let's say I call it T1, TK, then I would be basically creating this new amalgamated histogram by summing those little bins according to the threshold that I specified. So more generally, I have original histogram HI, I have amalgamated histogram HIT, and the way it looks will depend on the thresholds that I pick. Now for CGM, everything is integers, but you can think of it as a continuous relaxation. So you don't have to have an integers, and it's basically will be integrating your original distributional histogram for the regions corresponding to the thresholds that you select. All right, so let's me maybe give another illustration. So now I'm just talking about general thresholds, so general numbers. Suppose I give you numbers T1, T2, T3, T4. This is my original histogram. This is what it means to amalgamate it based on this given thresholds. I'm basically combining the bins up to T1, that I'm combining bins T1 to T2, T2 to T3, T3 to T4, and I'm creating this new simplex, which is this reduced or amalgamated representation of the original histogram. And I'm using always the same thresholds across all patients. That is what is currently done. But I'm questioning what is the value of those thresholds. Is this really 70, 180, or should it be something different? All right. So how can I figure out what those thresholds should be? For this, I need to have some measure of what does it mean for them to be optimal. And this is maybe a very intuitive idea. So I have my original histogram HI. Given thresholds T, I have my amalgamation HIT. And these are the two types of loss functions that we propose to consider. One is the distribution preservation. This is probably the most intuitive one. So I want my amalgamated histogram to look as similar to be as similar as possible to the original one. And then I look at the average of those distances over my subjects. The second one is a little bit more complex. And the second one, the idea is rather than preserving all the features of the original histogram as much as possible, I'm more focused on preserving those features that differentiates the histogram across each other. So the idea is that if the original histograms were far, I want the amalgamated histograms to be far. If the original histograms were close, I want the amalgamated histograms to be closed. So I want those distances to be preserved. And then once we have this notion of the loss, then we can think of the problem of threshold selection as the problem of selecting those cut points so that the loss is minimized. So we control how many cut points we need to select. That is total K. So coming back here, I have 1, 2, 3, 4 cutoffs. So I'm saying I want to find 4 thresholds. And this number 4 came from the scientific rationale because that's how many they use for CGM. But rather than using 54, 71, 80, and 250, I wanted the data to tell me what those 4 numbers should be so that I either preserve the original distribution as much as possible or I preserve the distances as much as possible. And then the question becomes, what is the right distance? Because we're looking at the histograms, which are distributional representations, we're going to work with the Wasserstein distance. It's an increasingly popular metric between 2 probability distributions. It is an optimal cost from transforming one distribution to another. So here's a quick illustration. Suppose F1 is a density for one distribution and F3 is another density. Then to go from F1 to F3, I'm basically going to be moving this peak from left to right. Whereas if I were to take the Euclidean distance between F1 and F3, I would be getting a bimodal distribution. So the Wasserstein distance preserves the geometry of the space. And it also, in our case, allows to account that we're sort of amalgamating this neighboring bins of the histogram. So more specifically, if you have one-dimensional distributions, then the Wasserstein distance can be expressed directly through the quantile functions between those two distributions. So it's a squared distance between the quantile functions, which means that if we have the quantile functions, then we can compute the Wasserstein distance directly in a univariate case. So coming back, we have our empirical original histograms. So which means we have empirical quantile functions. So we have those available for the data that we have. So the question then becomes, once we amalgamate the histogram, so given a certain thresholds, we could construct this representation. What is the quantile function corresponding to this amalgamated histogram? Because we want to measure the distance between those two, we want to use Wasserstein distance because we're comparing basically distributions. And the question then only becomes, what is the quantile function corresponding to this amalgamated thresholds? And this is where we view the histograms as a locally constant densities. So this is a toy example where I have 1, 2, 3, 4, 5, 6 bins. And let's say I'm thinking of amalgamating or combining those two pink bins. So originally for 6 bins, I had a quantile function that has 5 knots or 6 areas. If I view the histogram as a representation of a locally constant density, then combining or amalgamating the two bins is basically the same as linearizing the quantile function in the corresponding part. So in other words, it's as if I remove those knots and then just put a line between those two. In other words, I can think of my problem of optimal threshold selection or optimal histogram amalgamation as a problem of optimal piecewise linearization of the underlying quantile functions. And because it's linearization, I can express linearization in a closed form. So for any thresholds that I specify, I now know that that's a linearization of the underlying quantile function. I have a closed form expression for the linear interpolation, which means I can now write my losses directly through my original quantile functions and through my linearized quantile functions given the thresholds. So once I have those losses, the only part that is left is to figure out how to optimize. So how to find those numbers given the K. And there are several approaches we consider. I think the most straightforward is a greedy approach where you basically either perform stepwise aggregation, which means you start with all the bins and you just combine them one at a time in the greediest fashion possible with respect to the loss. Or you can start with one bin and split. In simulations, we find that splitting is highly suboptimal. So if you go this route, aggregation is the way to go. But fortunately, you don't even need to do the greedy approaches. It turns out that joint optimization is possible, and you can use a family of algorithms called differential evolution. It sounds fancy, but it's really just a Python function, and it allows you to solve this optimization problem jointly. We also have considered semi-supervised generalization. So this is the setting where, let's say, I say I want 4 thresholds, but I want to fix some of them at particular values. For example, let's say in CGM context, 70 is really important for me. I have a scientific rationale for why I really want to see 70, but for the other 3, I don't know. So you can do that too, which is you can fix some of the thresholds and optimize the others. All right, so this was the method part. So let me show how this works on the data. So this is what we have considered. We considered 2 data sets. One were subjects without diabetes and one was subjects with type 1 diabetes. We specifically picked 2 data sets that will represent 2 different populations. because we wanted to see how the thresholds may compare. They both have same CGM, Dexcom G6, which had 5-minute measurement frequency. And the both data sets are public. We actually have a large public repository of CGM data sets if you are interested, so you can reproduce these results. And we had two goals. First, we wanted to perform analysis separately for each data set. So we wanted to apply our approach to have 4 thresholds selected. Consensus of those, we wanted to find 4, and we wanted to see how they compare to consensus, as well as how to compare across the data sets because they represent different populations. In this case, we use distribution preservation. So we just wanted to match those original distributions as close as possible. In the second goal, we combined 2 data sets and we used the distance preservation. So we looked at preserving relative distances across. And again, we wanted to find the data-driven thresholds and see whether data-driven thresholds have higher discriminative power compared to the traditional thresholds. So those are the results on the subjects without diabetes. So here I'm showing you the empirical quantiles, which is the original full distributional representation of 168 subjects before any thresholds were applied. Then I show you what is basically the approximation of that is with the thresholds that we have selected versus the approximation with the traditional consensus thresholds that are currently used. And for this data set, I think it's very visually clear that the data-driven thresholds give you a much better approximation of the data than the traditional thresholds. And this is somewhat expected result because, again, traditional thresholds are designed with type 1 diabetes people in mind. Here, I'm using those thresholds for population that has no diabetes. So you can see that data-driven thresholds are much lower. They're going into the lower glucose values because this is where the subjects differentiate most. And from the loss perspective, it's a huge, huge improvement. So we evaluated what is this loss on the consensus thresholds, what is the loss value that we got with our thresholds, and it's just huge difference in terms of loss. When we look for type 1, we still see a difference, but here the difference is less pronounced. And again, this should be not surprising because the original thresholds were developed for type 1 population in mind. But the biggest difference we see is that our data-driven thresholds are higher. They tend to separate more in a higher glycemic range, where the consensus thresholds are more on the lower range. And in particular, these thresholds are higher than they were for population without diabetes. And again, this is reflective of the fact that people with type 1 diabetes have higher glucose values, so their quantiles go much higher. All right. This is what we got when we applied it across both data sets. And this is, for simplicity, just using 2 thresholds rather than all 4. So what I'm showing you here is the 2 data sets are separated left and right, and that there is a bar plot. So each vertical line is one subject, and they're just sorted according to the purple, which is the lowest threshold. So these are all people without diabetes, and then you have people with diabetes. And basically, hopefully what you can see visually, and then I have some numbers to convince you, is that if we use the data-driven thresholds of 149 and 255 to 56 to make it include in the upper level, visually those two are very different. And while they're still different at the traditional thresholds, you don't see as much difference in the lower part, and the higher part still overlaps. To kind of help you demonstrate it from a different perspective, so these thresholds create 3 number summaries. So in the data-driven thresholds, we basically had percentage below 149, percentage between 149 and 256, percentage above 256. For traditional, You get free summaries as well. It's just where your threshold is different. We used each of those individually in a simple logistic regression, trying to discriminate people from the first data set from people from the second data set, which is very easy because we're trying to distinguish people without diabetes from type 1, but we're trying to assess whether our thresholds are better at doing this than the data-driven one. And this is the results. So here you have our thresholds versus consensus thresholds. For each threshold, so treating it as one summary at a time, you do the logistic regression trying to separate. You see what the accuracy is, and you see what is the boundary. So for example, here, what this says is that if you spend 86.7% or more within this range, you're 100% accurately classified as a person without diabetes. And if you're spending less than that, you're 100, like it's 100% separation between those two data sets. Everyone who has no diabetes in this data set spends 86% or more below. Everything with diabetes spends less than 86% there. Similarly, we have 100% separation for the second threshold. So here with people with diabetes, they're 13.7 or less. People without diabetes are 13.7% or less, people with diabetes more. And then we have almost perfect separation in the third one. For comparison with traditional thresholds, you have very good distinguishing of the two at the higher levels, but your lower threshold is not that discriminative. And you can see this visually if you compare the purple between those two plots, they look very similar. So it's harder to separate based on that. So to conclude this part, we introduce loss-based threshold optimality accounting for the distributional nature of CGM data. Our loss is in particular built using distributional distances. It allows us to produce summaries that are still easy to explain and use in standard downstream tasks. It's unsupervised, so you're not gonna get overfitting. You're not gonna worry that you selected that threshold just because it's very well associated with your outcome. Those are unsupervised losses. And because we have this semi-supervised extension where you can fix some threshold values, we can enforce the hierarchy across levels. By default, the hierarchy is not necessarily enforced. The choice of the number of thresholds currently is driven by scientific knowledge. I think whether you can optimize that in some way would be similar to like how do you select the number of components in principal component analysis. I think there's some similarity here. Supervised context may be of interest. And then we saw that the thresholds sort of tend to select higher values. For CGM, we may be more interested into adjusting for the lower values ranges as well. And these are the references for this part of the work. Before I switch to the next part, I just wanted to see if there may be any questions or clarifications that I can answer. If not, I'm going to move to the second part. And this is where I'm going to talk about coming back to this distributional representation, but how I can use this full distributional representation as an outcome. And through this representation, see better how different covariates can affect glucose values. So this is based on those two works that you can find on arXiv. And this work has been driven by Alexander Coulter, who is a PhD student at Texas A&M. And I would like to also acknowledge my collaborators, Naresh and Nisha, who are experts in sleep apnea. And I'll tell you about that as a motivation for this work, as well as Rebecca Lee, who is also a PhD student at Texas A&M, and she helped a lot with the software. So the motivation for this work is we're going to go a little away from diabetes and then come back, and we're going to talk a little bit about the obstructive sleep apnea, which is a breathing disorder where when you sleep, you get distraction to your oxygen. And the reason it's going to be important for us is it turns out that the sleep apnea and diabetes often co-occur. In fact, there's an estimated prevalence of 54 to 86% among patients with type 2 diabetes. I believe that prevalence may actually be similar in type 1, but because type 2 is much more prevalent, we sort of know more about type 2. And it's not very well understood how sort of those comorbidities interact. So if you have a person who has both type 2 diabetes and they have a sleep apnea, I kind of one of the question is how would the fact that they have sleep apnea potentially affect their glucose control, especially since for the sleep apnea, we have several treatments that are available. So this particular data comes from the HYPNOS study, which is a study of around 200 patients who can currently have type 2 diabetes and obstructive sleep apnea. The glucose levels were measured by Dexcom G4 CGM, so it's an older CGM, it's an older study, but it's still 5-minute measurement frequency. And we had a lot of clinical information available, such as the age, BMI, the level of severity for the obstructive sleep apnea, diabetes medications, etc. And in this case, we really wanted to quantify how the baseline values of these variables potentially affect the glucose levels, and we wanted to use the data from CGMs as an outcome. And this is kind of some examples of what the data look like. This was full 24-hour profiles for 4 selected subjects from the study. So again, you can see that this is 300, so a lot of them run pretty high because these are people with type 2 diabetes. You don't have this clear free peaks, so you don't really know when they ate or what happened. I'm showing the time where they slept in blue. And again, it's sort of all over the place. There's high dependence of environment. And there's approximately 11 days on average that we have for each person. So it's a lot of data. And I'm showing you this because functional approaches are often used for wearables data as well. And what I'm trying to demonstrate here is it would be really hard to sort of align these profiles by time because peaks happen at all kinds of different times. We don't have a lot of information on what they did. And so this is where we are going to look at the distributional representation again, where we're collapsing this by time and we're looking at the full distribution of glucose values over the time. And I do want to acknowledge that I'm definitely not the first in the context of CGM looking at the distributional representation. And this idea was first popularized by Marcos Matabiana in this paper, calling it glucodensities. So again, the idea is this is our 70 to 180 that I told you before. This is the full distribution. And in this case, rather than trying to find an optimal threshold or use the threshold, we will try to work with that distribution as a whole. All right. So again, we have a lot of observations for each subject. We can construct empirical quantile function for each of these people, which allow us to balance more simplistic measures on full data. It does ignore the local time information, but it captures all the thresholds that are currently used. It captures the mean, it captures standard deviation. And by working again with this empirical quantile function, we're also not going to assume that this is like a normal distribution or some other distribution. We're just going to work with this distributional object as is. And we want to understand how 30 plus covariates that we have in this data set affect this distribution. We want to do variable selection because we don't think all of them have an effect. And we also want to quantify the size. We're going to again use the Wasserstein distance that I talked about before. So just as a reminder, again, it's a squared distance between quantile functions in a univariate case. And again, to remind you, it's better than Euclidean because if you had two densities, red and blue, the Wasserstein average will be green, whereas Euclidean average will be purple. So Wasserstein, again, preserves the geometry of the distribution space. All right, so how are we going to do this? This is where we are going to take ideas from Peterson and Miller work on Fréchet regression. So we have our quantile functions. So this is our Wasserstein distance. And we can find the average quantile function of Wasserstein barycenter by calculating the quantile that corresponds to the average distances. So the way we can introduce covariates and make it a regression problem is by basically weighting these distances. So we have our n subjects. Each one of them have some covariates, x, and now we have a new subject, x, that comes in. So the way we can think of creating the quantile function for that new subject, x, is by weighting the distances and doing this weights relative to how similar the subject x is to each of the xi's. So for each person, you have the covariates xi, you have your new covariate x at which you want to predict your quantile function. You look at the similarity of the weights between x and xi, and then you find this quantile function by sort of upweighting the distances to the quantiles that are most similar and downweighting the ones that are less similar. So it's an extension of an average that accounts for the covariates. So this gives you a fitted quantile value at a given covariate x. And the covariates comes within the weights, and then the object has to be a quantile function. So everything together, because our Wasserstein distance is just Euclidean distance between quantiles, you basically formulate this regression problem as a problem finding quantile function at a covariate x by averaging across quantile functions in your sample with the weights that look at the similarity between covariates of each sample and your covariate. And then you have a monotonicity and support constraint because you want a quantile function. The support constraint comes in because glucose values from CGM are only between 40 and 400. So it only makes sense to output quantile function that is between 40 and 400. All right. So this is most mathematical slide, so bear with me, but we're gonna come to the application next. What we want to do here is we want to do variable selection. But currently, when we look at those weights, we look at the similarity between vector of covariates xi and our vector x for each of the coordinates. So the way we can introduce variable selection within this framework has actually been proposed by Tucker, where we introduce an allowance vector lambda that modifies the weights. And the basic idea is that if a particular coordinate of this lambda is 0, then we eliminate that covariate so that covariate effectively doesn't enter the weights. And otherwise, it does enter the weights. So it's, you can think of it as a way to adjust the weight vector. So it only uses the subset of covariates rather than all of the covariates. It's very similar to lasso ideas in linear regression. So here is an illustration where if you have lambda 1 equals 0, lambda 2 equals 1, you're basically removing the first covariate and only getting the first one. So once we have these allowances, basically we can predict the quantile. So this allowance vectors tells us which covariates to pick, which to discard. Based on those covariates, we can construct the predicted quantile functions. And then if we want this allowances to be selecting only a small subset of covariance, we sort of control the total allowance, the total budget that we give it. In other words, we now have a more complicated optimization problem where, given this total allowance, which you can think of it as a tuning parameter that controls the model complexity, we are going to build the individual allowances vector that will tell us which specific covariates we need to select. And then we weight it within this regression problem, and that allows us to do quantile regression with variable selection. And even if you didn't follow the math, that is fine, because the biggest thing I hope to convince you is that this is not a straightforward optimization problem. This is actually an embedded optimization problem where you have to specify this allowance vector, you have to find the lambda, but then for a given lambda, you have to fit a quantile. So you kind of have this back and forth going on, and it's slow. And then because of how this is formulated, you may imagine that trying to do inference or providing some kind of asymptotic guarantees is going to be difficult because it's very difficult to analyze this object. So what we really wanted to do is kind of make, sorry, let me just tell you again where the computational bottleneck is. This is basically what we want to optimize. So we have the weights, which allow us to select some of the covariates. How much we select is going to depend on tau. And then once we have the weights, we need to find fitted quantiles at those weights. And then we find the best lambda so that the fit is the best. And this is very, very slow. And it's very, very slow because you, move one lambda at a time to minimize the subjective. The gradient is difficult to compute. The simplex constraints are difficult to work with. So there is sort of a lot of challenges. And then we don't have inference because it's very difficult to analyze. So what we really wanted to do is to make this fast, not just because we wanted to apply it to our data, but because by making it fast, we can then use resampling techniques to do inference. So we don't need to sort of look at it from the asymptotic perspective. And Alex came up with this brilliant algorithm where basically from optimization perspective, we change from the simplex to the sphere. We can study the optimization problem more to derive gradient and Hessian. And then when we do the updates, we move on the sphere. So we're sort of preserving the geodesics rather than moving out and projecting back. And that just, all of this together makes it much, much faster. So just to show you like how much faster, so black is our algorithm, gray is previous algorithm. Notice the scale of the y-axis. This is just for 10 and 20 covariates. We can do it with 1,000 covariates. And it can run on a laptop. It's gonna be really, really fast. And the reason this is so wonderful is because it allows us to do variable selection in a way that is more principled than just trying different taus and figuring out which model is the best. So specifically, this is the problem that we can now solve fast, where we have an allowance vector tau, which sort of controls the model complexity. The larger is the tau, the more variables you will have. The smaller is the tau, the less variables you have. And then given the tau, it gives you the specific variables. And rather than finding what this tau should be, what we are going to do is we are going to take a range of tau values, so a range of models from least complex to most complex, and we are going to do a lot of subsamples from our data, and then we are going to look at the stability of selected covariates across those subsamples. So this is the idea of stability selection. It's traditionally used with linear regression and lasso, but we can use it for our case of distributional regression here. So basically we look at B subsamples of roughly half the size. We look at this grid of model complexity values and we fit the model on all of those subsamples across the grid. We'll look at what is the proportion of time that the variable is selected across subsamples. So those are those lambda values that we estimate. And then we define the stable variables as those that are selected more than a threshold. And this is expected to control the number of false discoveries based on the previous work. So by doing variable selection in this way, we have some idea of what is our false discovery control. So we have some inferential properties. All right, so back to the hype now. So if you fall asleep a little bit of optimization, let me show you how this works. So as a reminder, we had 200 subjects. Here we have 34 covariates. And we're discretizing quantiles at 100. So that just means that when we compute those distances between quantiles, we're working with like 0.01, 0.02, 0.03, and so forth. This is the characteristics of the data set. So this is people with type 2 diabetes and obstructive sleep apnea. So you can see that they are older, they're balanced in terms of gender. The A1C, which is the measure of the basic glucose control, lower is better, has quite a range. 11 is really high. These two medications, bigonide and sulfonylurea, are very commonly prescribed diabetes medications within type 2. You can see that most people had bigonide, some people had sulfonylurea. They are not mutually exclusive, so you can have subjects with both. And then the other three are measurements related to obstructive sleep apnea. So they are measurements related to the oxygen saturation during the night, and there was 5 measurements total. So the first thing we did is we actually looked at the correlations across our 34 covariates. And for those of you who are familiar with lasso instability selection, you know that when you have covariates that are highly correlated, the variable selection could be unstable. So we wanted to make sure that this is not the case. And this is the heat map of the correlations across our 34 covariates. And you can see that there is a cluster over here that is more highly correlated. And those were actually 5 different measurements related to oxygen saturation, related to sleep apnea. So what we did is rather than using those 5 original ones, we did principal components on those 5. So we have 5 orthogonal measurements, and we did variable selection with respect to those 5 principal components. So we knew the correlations was not an issue. So to do this variable selection, we use a grid of 40 tau values. So as a reminder, this is the measure of model complexity. So basically ranging from models that have almost no covariates to the models that have a lot of covariates. And then for stability selection, we used 50 subsamples of our data. So we took 50 samples and we did 40 tau values, which is 2,000 evaluations of that objective, took 4 minutes on a laptop. So that just tells you this is very fast. This is what those stability selection paths look. So again, this is the tau values from a simplest model to the more complex model. The stability measures proportions that the variable was selected. So you have 34 lines corresponding to 34 covariates. The gray line is sort of mean selection probability, which the more complex is your model, the higher is the average selection probability because you're always going to select. And the purple line is a threshold from the previous research on what should be the threshold for selection. And the 3 highlighted trajectories are the 3 covariates that at some point got over that threshold, which is HbA1c, sulfonylurea, and one of the principal components associated with the oxygen saturation. And you can see that it was more borderline, but it still hit the threshold. And we actually found from the simulations that that threshold is quite conservative. So we are pretty confident in those results. So basically what this tells us is that out of the 34 covariates, we found 3 that have evidence of association with a glucose profile. So A1C is sort of a self-fulfilling prophecy because we had the values of A1C before the CGM was put on. So this is the average glucose values for the preceding 2 to 3 months. And then we look at the next 10 days of glucose values. We would expect this covariate to come in. Sulfonylurea is the oral diabetes medication, and then the PC2 is the second principal component with oxygen desaturation. So to figure out what is the effect, what we basically did is we used this distributional regression framework to construct fitted distributions with different values of those covariates and see how the fitted distributions change as a result of change in those covariates. And this is the results. So I'm going to start with A1C first. So I transformed the quantiles to densities because it's a little easier to explain. But basically for the bottom range of A1C, you see that the glucose values tend to be lower as you would expect and tighter. And as the A1C grows, that are sort of those grays transforming into the purple, your glucose values gets higher and also more spread out. And again, this is a self-fulfilling prophecy because A1C was measured just before we put the CGM in, so we will hope to see this. What is really interesting is the sulfonylurea, and this is one of the supports for the distributional representation. So you just have yes or no, people on sulfonylurea are in purple, no sulfonylurea is blue, and you can see that the center is the same. So if I were doing this analysis on the means, I wouldn't see this effect. But the spread is different. So basically what this tells you is that for people who have sulfonylurea, it is associated with variability. It is associated with a spread, but not with the overall levels, not for the median. And you can capture that because we're looking at the distributional effects. And then when we look at the PC2 oxygen, it's the smallest effect, and you see that there's a little bit of a shift to the higher values of glucose when you go from the top to the bottom range. So it's also associated with overall glucose values. I'm going to skip this interpretation and sensitivity, and I think I'll just go to the summary now just to show you that in this work, we demonstrated that distributional regression is a powerful alternative to linear regression. And what I mean by that is that it really allows the detection of effects beyond the mean, as we have seen with the sulfonylurea, where we detected the distribution is wider rather than it's shifted. We've developed faster algorithm. That was our primary contribution. But because we have this fast algorithm, subsampling is not an issue. So you can do uncertainty quantification using subsampling approaches. And from optimization perspective, the key ideas were to take advantage of optimality conditions, try to do gradient updates that preserve the geometry. And the key findings were that we were able to find that oxygen saturation does have an effect, and it actually has an effect more in terms of variability in those profiles, oxygen profiles overnight, rather than the overall levels. And we're curious to explore this more in future studies. And then from the distributional representation perspective, in the future work, we want to look at how do we do repeated measures? Can we do interactions in addition to main effects? And then, as I told you with CGM, whenever we do distributions, we collapse local information. Is there a way that we can incorporate some of that local information back? And this is the reference for this second part of the talk. We have a very fast R package on GitHub if you want to use it up. And with that, I think I will stop. And thank you very much for your attention.